Thank you very much, Erin, and thank you for your flexibility mid-presentation. Okay, we did receive one question, and I'd encourage anyone else who'd like to ask Erin a question to put that in the Q&A box. But this question, Erin, is about tissue expression for a particular gene, and if in fact it's in conflict with the condition that you are analyzing the gene for. In that case, how would you factor that into your analysis?
So that is a great question, and it's one that we think about a lot within the ID autism group and the epilepsy group, which are the two groups that I'm affiliated with, just because so many genes are expressed in our tissue of interest, which is the brain. And we've actually decided as that group that because that particular finding in our particular tissue of interest is so nonspecific, we don't count that as information for a gene disease association, just because so many genes have that. It's not very special, it's not very specific, it's not telling us anything. You might feel completely differently if you were part of a group assessing conditions involved in other tissue types where expression is a little bit more limited and it is more indicative of a role in disease. I wouldn't necessarily say if something wasn't expressed in your, or you didn't know that it was expressed in your tissue of interest, I wouldn't necessarily call that anti-evidence, just because we know there are limitations in the resources we have available for tissue expression. It just may be that we haven't demonstrated enough, or we don't have the resources to show that. So I think that's just a domain-specific issue where each domain might consider things differently depending on the resources they have available.
Okay, great, thank you. And another question just came in that something to add to a lecture like this would be walking the participants through the scoring details with a case study. So I know we may not have time now, but we do have resources on our website that you could maybe point participants to.
Yeah, absolutely. And if you guys have specific examples that you would like to see walked through, that could be something that we add to a future tab on our website that we have for this workshop, to make things a little bit more accessible for you.
Okay, and we don't have any other questions right now. I'd encourage people to continue submitting because we do have some time before the end of the workshop. But Erin, with the extra time, would you like to walk the group through just how to navigate the ClinGen website and looking up some of the gene curation results that are available from our...
Yeah, absolutely. Can you confirm that you see the screen?
Yes, we see the homepage.
Okay, great. So I went through this very quickly in the slides just because I wasn't sure how much time I would have, but thankfully that we have extra time and we can take a little bit to look at this live just so you can feel comfortable using the ClinGen website. So this is our website. It's at clinicalgenome.org. And here you can access information from all of our different curation activities. Since we're focused on gene disease validity right now, I'm going to focus on that in this demo. If you are interested in just kind of seeing what we have for gene disease validity, you can click on this tab and click on all curations. And it'll take a moment to load, but then it will load all 1,200 curations that we currently have. Within ClinGen, we are doing gene curations on almost a daily basis. So anytime any one of the groups completes a curation and publishes it to our website, it will become available here immediately. So it's not that we hold data for a year and only do releases once a year or anything like that. The website is continuously being updated, so I encourage you to check it frequently because things are always changing. So if you want to see the list of everything, that is how you find it. If you are interested in downloading this list, you can click on this button here, which looks like a line with an arrow coming out of it, and download it into any one of these formats of your choice. You can also do a few different things within the columns. You can sort any column by highest or lowest value just by using the arrows. And you can also look at adding additional columns that we may not already be displaying. So for gene curation, if you are really interested in knowing what the HGNC ID is, for example, or the Mondo term is for this particular display, you could add those columns here. So I'll just add this one. And if there was a column you weren't interested in, you can take it away using this button here. So that's what I've done. Another feature that you might find useful is this one that looks like a magnifying glass. This allows you to search within any of the columns for anything that you might be interested in. If your box does not have one of these little arrows under it, that means that you can search free text within that box. So for example, under gene here, if I just typed in ZN, I will get back all 3 genes that we have curated that have ZN within their gene symbol. The boxes with the arrows will show you you have certain choices with which to search. So if I drop down this mode of inheritance, I can choose any of these modes of inheritance that I might be interested in. So I'll choose X-linked. You can also choose which classifications you're interested. So if you wanted to know, hey, how many definitive X-linked curations are there? And we can see that there are 85. And then within this last evaluated column, you can search by year if that was something that you're interested in. So I'll choose 2018. And you can see that narrows our list down to 34. Again, if you used a bunch of these features to kind of hone in on a set of genes that you were particularly interested in, you can also download that list. So like this download button doesn't just apply to the full data set. You can filter it in however you like and then download that. The other thing that I just wanted to point out that you can do for gene curation is to look by expert panel. So if you are really interested in a particular disease area and just wanted to hone down the results to what we might have in that particular area, you can come to this page and see a listing of all of our GSEPs or gene curation expert panels and peruse the data that way. So I will just pick fatty acid oxidation disorders and I can see all 28 of their curations there. That might be useful to you, again, if you're interested in a particular disease area and there's no kind of overarching disease term that you could use to effectively search. You know, so if I was interested in fatty acid oxidation disorders, I can't just put in the phrase fatty acid oxidation disorder and get all these, because you can see those words don't appear at all in any of these disease terms. So it would be difficult to search in that way. So we hope that being able to search by panel allows you to get at that information a little bit more clearly. And then the final thing I'll say about this is you don't have to look at all of these lists if you just have a particular gene that is your favorite gene. Mine is ZEB2. You can come up to the top and search it directly and get to its specific gene page. And here you can see we've evaluated that particular gene for gene disease validity and dosage sensitivity, and you can access both of those that way. I'll stop sharing and see if any additional questions have come in.
Thank you, Erin. So one question about genes to be reviewed. Can you request a gene to be reviewed by?
Absolutely, you can. So let me share screen again. So at the bottom of each page, it's kind of hard to see, but I'm glad I'm able to show it to you here today. There's this little green button called Send Feedback. And if I click on this button, it just says, you know, keep in mind a few caveats. You know, this is not intended for diagnostic use or medical decision making without review by a genetics professional, etc. Just say that you have read that. Then it will ask you just some information about yourself in case we need to contact you back to ask any follow-up questions. And then you can indicate what kind of feedback you're providing. So if you wanted to request that a particular gene be evaluated for gene disease validity, you would type that. Please review ZEB2. I'll just put this as a test for when I get this email later. I can remember that. Tell us you're not a robot and submit the feedback. Oh, I didn't put in my name. Anyway, that's how you can very easily request a gene. If you can't remember that, you can just simply email us at clinigen@clinicalgenome.org and we will get that request into the appropriate GSEP.
Okay, and next question. What about gene curation for novel gene with no available information? Do you look to homologous or orthologous genes or something else?
No, I mean, for gene disease validity, again, because it's so important for kind of the downstream consequences, you know, if we incorrectly assess the validity of the gene, then, you know, we may subsequently incorrectly assess the variants that we find in that gene. So we really are looking for evidence for that specific gene. If there's none, we do have a classification called no known disease relationship. But typically, for ClinGen's purposes, we are evaluating genes that have at least some information. There was some reason why somebody said at some point in time that this is a gene, a candidate gene, and so we kind of take that information into account. Again, like I said, we have the no known disease relationship category if there's truly no genetic evidence. And then we do have some cases in which, you know, there had been some information at some point in time, but we have ruled it out, or we would call those either disputed or refuted. Within the no known disease relationship category, if there was, you know, if, for example, that gene was purported to be a disease gene because of functional data, like perhaps there was an animal model that suggested there may be a relationship to disease, we do have a flag available to say, hey, this one is classified as no known disease relationship, but there is an animal model available.
Okay, thank you. That finishes up the questions that we had received on gene curation, so thank you.
Well, since we do still have a few moments, I might turn it back over to you, Danielle, because I did mention in the talk very, very briefly the resource matchmaker exchange. And I think, you know, since we have time, if you wouldn't mind just showing everyone that website and explaining briefly what that is, just because I think it's a really good resource and I want to make sure everyone's aware of it.
Sure thing. So I am going to share my screen and take you to matchmakerexchange.org, which is the website that Erin had described in her talk. And can everyone see that?
Yes.
Thank you. So Matchmaker, as was mentioned in different sessions, is a service so that if you do have a candidate gene in a patient that you believe could be causing their suspected genetic diagnosis, you can take high-level information from that case, so the name of a gene or several genes, and some of the high-level phenotype or condition information that describes their presentation, and enter that into a database that can connect you with others who have similar information on unsolved cases to see if you can find a match and build evidence towards a gene disease relationship by accumulating these cases. So one thing I'd like to point out is that Matchmaker itself, if you visit our website, is not the query service, but it brings together all different databases, currently 8 different databases globally that accept cases and then share with each other these cases to find matches. So if you're hearing about this and you think this is something that may be a useful tool in your clinic or laboratory, I would encourage you to go to matchmakerexchange.org. Under how to get started, and I have a case to match. You can learn about the databases that we have available that are currently matching these unsolved cases between researchers, laboratorians, or clinicians, and learn more about them. Some of them are closed to research groups. Others, such as Gene Matcher, are freely and publicly accessible to anyone who has a case and is willing to enter information. So I encourage you to visit here, learn about the different databases, then you'll go to those databases and establish an account with them to submit cases to. But once in there, you'll be able to match your query against cases in all of the connected data sets. So Matchmaker Exchange is really just describing the connection between all of these individual data sets that serve to do this genomic matchmaking between unsolved cases. There is a contact us button that goes to info@matchmakerexchange.org, and we're happy to walk you through that in more detail if you think this is something that could be of use in your clinic or lab.
Now, you said this really briefly, Danielle, but are there any considerations people should keep in mind as they choose which node they might want to participate in, or does it not matter? Like any of them would work in any circumstances, or are there things to consider?
Yes, so First, accessibility is certainly one of them. So when we go to the exchange participants, we have different descriptions of the type of data that the databases collect and what they match on. So you can find something that aligns with the cases that you have in front of you and also the permission level you have to share that information. So some make matches just based on a gene and phenotype. Others allow you to search more specifically with additional phenotype information or on a variant. So it really depends on what you're looking for. And again, I said some are closed to certain research groups. So if you're not a part of that research group, that wouldn't be the best database for you to start, but several are available to anyone who makes an account, including GeneMatcher. So that's a good start for someone who's not associated with a research project that currently has an established matchmaking database or node.
Yeah, and that country in the parentheses, that's just the country of origin for the tool. That's not saying like only people in that country can participate, right?
Correct. So that is a great question. These are just where they are based, but they all accept cases if they're open from across the world. So this is just where they sit.
All right. Well, thank you so much, Danielle.